Repeated immunizations lead to increasing affinity of antibody owing to somatic hypermutation and selection by antigen in germinal centers. Upon re-exposure to the same antigen, a secondary immune response will ensue. In some ways, this resembles a primary immune response with the initial proliferation of B cells and T cells at the interface between the T and B cell zones. The secondary response is characterized by early and vigorous generation of plasma cells, thus accounting for early profuse immunoglobulin G production. Some B cells that have not yet undergone terminal differentiation can migrate into the follicle and become germinal center B cells. There, these B cells enter a second proliferative phase, during which the DNA encoding their immunoglobulin V domains again undergoes somatic hypermutation before the B cells differentiate into antibody-secreting plasma cells. The antibodies produced by plasma cells in the primary and early secondary response have an important role in driving affinity maturation in the secondary response. In secondary and subsequent immune responses, any persisting antibodies produced by the B cells that differentiated in the primary response are immediately available to bind the newly introduced antigen. Some of these antibodies divert antigen to phagocytes for degradation and disposal. If there is sufficient pre-existing antibody to clear or inactivate the pathogen, it is possible that no immune response will ensue. However, if there is a trace excess of antigen, B cells whose receptors bind the antigen with sufficient avidity to compete with the pre-existing antibody will take up the uncomplexed antigen, process it into peptide fragments, and present these peptides bound to MHC class II molecules to armed helper T cells, surrounding and infiltrating the germinal centers. Contact between the B cells presenting antigenic peptides and armed helper T cells specific for the same peptides leads to an exchange of activating signals and the rapid proliferation of both activated antigen-specific B cells and helper T cells. Thus, only the higher affinity memory B cells are efficiently stimulated in the secondary immune response. In this way, the affinity of the antibody produced rises progressively, as only B cells with high affinity antigen receptors can bind antigen efficiently and be driven to proliferate by antigen-specific helper T cells. And that's repeated immunizations lead to increasing affinity of antibody owing to somatic hypermutation and selection by antigen in germinal centers. Have a great day.